Hey everybody, Mr. Murphy here. Today I'm going to be going over Chapter 10, which is all about going from DNA to proteins through the process of gene expression. In this chapter, we're going to go through a very brief intro to just genetic ideas, and then on to how we transcribe, translate, and then what happens after translation. The chapter opens with a question like they all do. Here it's how do antibiotics such as tetracycline target bacterial protein synthesis. So you can imagine it's going to be involved in the process either of transcription or translation. So we'll see a little bit more about that. The chapter opens with just sort of exploring the historical idea of the association between genes and proteins. Because remember, as we've talked about in class, there's a rich history of the step-by-step process in which we came to understand that nucleic acids were even a thing and that they were entirely responsible for the machinery that is produced inside of the cell in terms of the proteins and their various effects. So there's some examples given, one involving phenylketonuria, which is a disease which causes the inability, you lose basically a functional enzyme that can convert phenylalanine, which is one amino acid, into tyrosine, which is another, which can have pretty significant effects in terms of your ability to first off moderate the amount of phenylalanine that you have, but then also reducing the amount of available tyrosine. It's very treatable nowadays, but if you ever notice on the side of like different food packaging or cans of soda or something, it'll give a warning if it's something that a phenylketonuric needs to be concerned about because of the contents. Things that if you just have that enzyme, you don't even think about. So this example and others gave rise to basically this idea of one gene, one protein. And that has since been revised, and so we should understand our current understanding of this is that each gene codes for a polypeptide. It's a subtle change from thinking of it as a protein, but it's because we now know that proteins can consist of multiple polypeptide chains coming together in that quaternary structure of proteins. So that's a big takeaway from this opening section of the chapter. It goes into some cool examples, just sort of exploring the slight amino acid differences in different variants of particular genes within populations within or across different species. So this is exploring gene mutations and how the amino acids therefore change. All ideas that we'll dig into over the next few chapters. So in this section, again, in this chapter, the primary focus is on the main two steps of this, which is transcription and translation. And throughout this chapter, we're going to look at three different types of RNAs. Messenger RNA, which is the one we're probably most familiar with. Ribosomal RNA, which we're also kind of familiar with, although we don't often think about it as being RNA. These are the RNA components which come together to form the ribosome. And then the last one is transfer RNA, which is one that you have most likely heard of, which is responsible for the delivery of the amino acids during the process of translation. So all of this is going to be covered in Chapter 10, and that's it for this opening section. So next up is transcription.